Okay, good afternoon, students. We are learning or discussing regarding the cell cycle. That means how does cell complete its life? In the previous session, we discussed about the interphase in detail by considering the occurrence of molecular events, that is the G1 phase, S phase, and the G2 phase. And once cell passes through all these three stages, now that cell is said to be completely mature cell. And this kind of mature cell, now it gets ready to enter into the phase of division, stage of division. Okay, and cell during this cell does undergo either mitosis or meiosis. ಇದನ್ನು ನಾವು ಒಂದು ಎಕ್ಸಾಂಪಲ್ನ ತಗೊಂಡು ಕಂಪೇರ್ ಮಾಡ್ಬೋದು. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಯು ಪೀಪಲ್ ಆರ್ ಫೇಸಿಂಗ್ the academic course through semesters. In a single semester, first your job is to prepare for the examination through listening to the lectures, through reading the textbooks, referring the notes, different references, etc. Completely prepare ಆಗ್ತಿದ್ರಿ. Then it is just like interphase. Then once you complete preparation, the date will be announced and you will get ready to give the examination. Hanga cell completely interface ಮುಗಿಸ್ತು ಅಂದರೆ ಕಂಪ್ಲೀಟ್ಲಿ ಡೆವಲಪ್ ಆಗಿರ್ತದೆ, ಪ್ರಿಪ್ರೇಷನ್ ಮಾಡ್ಕೊಂಡಿರ್ತದೆ, ಆನಂತರ ಮುಂದದು ತಾನು ಡಿವೈಡ್ ಆಗಲಿಕ್ಕೆ ತಯಾರಾಗ್ತದೆ. ದಟ್ ಮೀನ್ಸ್ ವಾಟ್? Why does a cell divide? Because cell contains protoplasm, and the ability to divide is an inherent property of protoplasm in every living cell. Okay, and this is the way how exactly we need to have the background history about the cell life or life of a cell. And also, before learning in detail about a cell division or a division of a cell, whether it is a animal cell or a plant cell, broadly in every living multicellular species organism, the cells present are differentiated into two kinds. Widely present somatic cells, where their nucleus is in a diploid state by containing 2n number of chromosomes. Another type of cells, they are called as germ cells or the reproductive cells, where their nucleus is haploid by containing just a single set or n number of chromosomes. But now, based on the type of cells, we need to study the cell division and the type of cell division. Broadly, we identify the division of cell or cell division into mitosis, and another one is meiosis. Still there is one more that is called as amitosis, where it is not equal distribution of their cytoplasmic or nuclear content that is observed in case of unicellular organisms. We are now going to study these two kinds of cell division. First one is mitosis, second is meiosis. Irrespective of the type of cell division, usually a cell divides mainly under two steps. First step, that is called karyokinesis. Second one, that is cytokinesis. Karyokinesis is division of a parental nucleus into two daughter nuclei. Secondly, it is followed by cytokinesis, that is division of a cytoplasm, means division of a parental cytoplasm equally into its daughter cells. So whether it is mitosis or meiosis, cell has to undergo these two stages. Firstly, karyokinesis by division of nucleus. Secondly, cytokinesis by the division of cytoplasm. Okay, and now let us try to understand the detailed account of mitosis. See, first of all, mitosis is known to be equational cell division. Equational cell division. What do you mean by that? Whatever the number of chromosomes present in the parental nucleus and the content of the cytoplasm, both are equally distributed to their daughter cells. That's without any variation, either in the number of chromosomes or in the amount of cytoplasm. Equal distribution, if it is seen, then it is called as equational cell division. For your reference, let me tell you another one kind of cell division known as meiosis. It is not equational cell division, it is a reductional cell division. How is that? We will study while doing it. Now, today, we will do only the mitosis. Before knowing the detailed account of mitosis, let us try to understand the significances of it. What all is achieved by the cell by following the mitosis type of cell division? First and the foremost is mitosis helps a cell to maintain equilibrium between DNA and RNA. The amount of DNA and RNA to be present in a proper ratio in every living cell has to be regulated, and it is achieved by undergoing mitosis. Second significance, it is to maintain shape and volume, or we could say size. Maintenance of the size or volume of a cell is possible by undergoing mitosis. Thirdly, mitosis helps the cell to replace old, decaying, injured, dead cells by new one. What do you mean by that? Once any cell undergoes the aging process, it becomes older. As it grows, it becomes older. And such type of very old or completely mature and old mature cells, then decaying or injured cells or the dead cells because of the injury, should be replaced by the newly formed cells, and that is achieved by mitosis. Then fourth significance, that it helps a cell to mitosis allows a cell to initiate cleavage during embryogenesis. What is that? Once a cell undergoes, that is female gamete undergoes fertilization, that becomes a zygote. That zygote has to undergo the subsequent developmental stages by means of undergoing a process that is called as cleavage. But this cleavage is facilitated by the mitotic division. So zygote likes to undergo division, likes to undergo division. That's what we call it as cleavage. Fifth one, mitosis facilitates the growth of a cell, facilitates growth of a cell, and lastly it also facilitates the formation of haploid cells. They are nothing but gametes. So all these are the factors achieved by mitosis, achieved by a cell by means of undergoing the process called as mitosis. So mitosis is referred to be in generally as a duplication in the number of cells. Just a duplication, that is 1 into 2, 2 into 4, 4 into 16, 16 into 32. Like that, there is continuous duplication or multiplication in the number of cells. For that point, mitosis is only possible, a possible reason for a cell to increase the number of cells. Okay, and after knowing the detailed account of the significance of mitosis, let us try to know the different stages that occur during mitosis. This entire thing you could get as a question like write the significance of mitosis as a short note, that is for 5 marks. If you write this, that could suffice the requirement during the examination. And now we will start knowing about the mitotic stages that occur in a sequence, one after the other. As I said before, that is mitosis. The first one, that is, let me write it like this: P M A T. That refers to what? P refers to first one, that is prophase. M refers to metaphase. Then A refers to anaphase. Then T refers to telophase. Okay, and just I give you the meaning. Pro refers to, pro means before. Then phases means appearance. Similarly, metaphase, meta refers to after. Rest of the thing is same. Then anaphase, this is up. Sorry, ana refers to up. Up refers to the upward movement of the chromosomes during that particular stage. We'll see it later on. And lastly, telophase, that is telo refers to last or end, last phase. Okay, that is fine. Please focus your mind on this chart. I am going to refer for explaining all these 4 stages. Firstly, this is for your reference, for your understanding. This is a cell which is said to be completely mature cell. To say that it is mature, it must have completed the phase, that is interphase. So now the cell is having everything with respect to its cytoplasm and the nucleus with a duplicated number or duplicated amount of their components or the contents. Okay, now this is kind of cell, it is said to be diploid cell, means what? Inside the nucleus, the number of chromosomes to be present are 2n. Other jodi irbeku. It may be 4, it may be 2, it may be 8, it may be 6, it may be 10, like that. Sama sankhya bartade, that is a pair. So that is said to be diploid cell. Such a type of diploid cell, now it is ready to undergo the division. So as to undergo mitosis, it has to pass through these 4 stages one after the other. First prophase ಆಗಬೇಕು, ಅದಾದ್ಮೇಲೆ metaphase ಆಗಬೇಕು, ಅದಾದ್ಮೇಲೆ anaphase ಆಗಬೇಕು, ಆಮೇಲೆ telophase ಆಗಬೇಕು, and that will complete the division of nucleus. Okay, ಇಲ್ಲಿ ಬರ್ರಿ. So this is the image of first phase, that is called as a prophase. But early prophase, late prophase ಅಂತ ನಾವು ಹೇಳ್ತಿದ್ದೀವಿ. So second one, this is metaphase, I mean nucleus or cell in metaphase. This is anaphase, cell in anaphase, these two diagrams. And this one is a cell in the last phase, that is telophase. Adana amele nodoಣ. Firstly prophase. Prophase nalli what all happens? Let me tell you firstly the points that occur one after the other. Prophase anna complete ಮಾಡಲಿಕ್ಕೆ first and the foremost thing is that there is a condensation of the chromosomes. Please remember, or condensation of the chromatin material. Number 2 is the disappearance of the nucleus, disappearance of the nucleolus. Thirdly, disappearance of a nuclear membrane. Fourthly, there is a formation of mitotic apparatus or spindle apparatus. ಈ 4 event ಆಗ್ತಿದೆ. First one ನಾವು ಹೇಳೋದು ಯಾವುದು ಅಂತಂದ್ರೆ there is a condensation of the chromatin material. ದಯವಿಟ್ಟು ಒಂದು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಮಾಡ್ಕೊಳ್ಬೇಕು, ಡ್ಯೂರಿಂಗ್ ನಾನ್ ಡಿವೈಡಿಂಗ್ ಫೇಸ್ ಅಂದ್ರೆ ಇಂಟರ್ಫೇಸ್ ಇರಬೇಕಾದಾಗ chromosomes are not conspicuous, they are present in euchromatin state. While cell undergoing division, that euchromatin gets converted or transformed into heterochromatin. What do you mean by heterochromatin? Heterochromatin is the one where the chromatin material is found to be very much shortened, thickened and condensed. And such type of condensed chromatin material appears like clearly a chromosome. So here you could see the beginning of the anaphase where there is shortening and thickening and they appear conspicuously. And finally look at it in the late prophase. What you find is that each chromosome is having two chromatids, chromatids. This is one chromosome where 2 chromatids are there. This is another chromosome with 2 chromatids. This is another chromosome with 2 chromatids. And this is 4th chromosome with 2 chromatids. So total how many chromosomes are there? 1, 2, 3 and 4. Whereas each chromosome is with 2 chromatids. Both the chromatids are strongly held together by a primary constriction called as centromere. This is centromere, this is another, this is another, this is another. Number of centromeres is equal to the number of chromosomes. Whereas each chromosome is with 2 chromatids which are almost identical to each other. That's why they are called as a sister chromatids. These two are sister. These two are sister chromatids. These two are sister chromatids. Okay. Idu condensation ಆಗ್ತಿದೆ. Second thing, there is a disappearance of nucleolus. It begins to disappear. Finally, it is completely disappeared here. You all know that nucleolus is mainly containing the RNA and histone proteins. Okay, that disappears. Then thirdly, there is the movement of these chromosomes towards the inner side of the nuclear membrane, by keeping their centromeres towards the inner surface of the nuclear membrane or nuclear envelope like this. So it is by making the central space of the nucleus empty, and these are found adherent to the inner side of the nuclear membrane. This occurs and finally after that there is a wider movement of the chromosomes, wide gap in between them, and finally that nuclear membrane also disappears, and the last event is regarding the centrosomes or centromeres. I want to say they are involved in the formation of mitotic apparatus or spindle apparatus. What do you mean by that? First and the foremost thing is that for each cell we find what? Two centrioles, which centrioles are present in pair, but while undergoing division they are found duplicated, means two pairs of here one pair and here one pair of centrioles. They begin to radiate, they begin to produce the thread-like or fiber-like structures. They are nothing but microtubules made up of protein called tubulin in all possible directions. Please try to visualize, imagine that a cell is not square, cell. Imagine that here is a cell, it is a ball-like, or it is a square. You remember, it is having all the sides, and inside at the center this is all happening. Okay, these two pairs of centrioles begin to produce the fibers with respect to their own set, and they also begin to move away from each other so as to approach their respective poles. For each cell there are two poles. This is one end and this is another end, or poles. This is one pole and this is another pole. So one set of centrioles will remain here only with their respective fibers. They are called as asters or spindle fibers, and it also begins to move away from it and finally gets located here to the opposite pole of the cell, and it is also having its own set of fibers. So these spindle fibers are found like this, but their fibers keep on extending so as to have connection with these centrioles. Please remember, this primary constriction known as a centromere is in turn surrounded by covering that is called as a kinetochore, and this kinetochore and the centromere is rich in DNA material, and this kinetochore begins to produce its own kind of fibers, and they are also called as ester fibers. One side spindle fibers, and in between the centromeres of kinetochore of each centromere of chromosome produces fibers, and they together connect in the end of the late prophase. Okay, that's all about the prophase. What all? Prophase, during prophase number 1, condensation of chromosomes. Second one is disappearance of the nucleolus. Third is the disappearance of the nuclear membrane, and fourthly there is a formation of spindle apparatus or mitotic apparatus, that is with their the fibers or thread-like structures known as the esters or spindle fibers. Okay, and now it is now into the metaphase. Metaphase is the phase where the chromosomes are found to be aligned, arranged in a specific plane, and this plane in the middle is known as the equatorial plane, and the entire setup, equatorial plane with aligned or arranged chromosomes, is known as equatorial plate. This is known as equatorial plate. Please remember how it is possible for all the chromosomes to come and align in the equatorial plane. That is because of these spindle fibers. These spindle fibers come and bind with the fibers of kinetochore, which is present around centromere of every chromosome. Okay, here are 4 chromosomes. Here also you can see 4 chromosomes. Their kinetochore of their respective centromeres are connected with the spindle fibers. Like that, from both the ends, from both the poles, they form somewhat like a dome-shaped structure, and they are held together in a balanced way, or in a balanced manner, and this is called, they produce kind of tension. Okay, they produce kind of tension. And one more thing I want to add up, that around the centromere of each chromosome, what we find are the microtubules found to be more added from here. So this is called, here the spindle fibers, they get deleted and come and added to this centromere of each chromosome. So this is called plus end and this is called minus end. Plus end and minus end, they produce a kind of tension. So because of that force of tension, these all chromosomes are very much properly held in a proper plane or equatorial plane with respect to their kinetochores or centromeres. Okay? Just ಇಲ್ಲಿ ನಾಲ್ಕಿದ್ದು ಆ ನಾಲ್ಕಿದ್ದಂಥ chromosome arrangement ಹೇಗಾಗಿದೆ ನೋಡಿ. Then you ask me a question that what is that driving force making these chromosomes to move in this way? That is all believed to be because of the action of certain enzymes. They are called as mitochondrial enzymes, MPF enzymes, as well as tension forces. All these forces and the enzymes are solely responsible for the proper movement of these chromosomes, as well as the formation of these microtubules which are made up of protein. And also there is action of kind of fibers. They are known as tubulin dimers. Tubulin dimers here. Tubulin dimers. Tubulin dimers are actually present here and they are removed there and they get added to this point with each chromosome, and addition of tubulin dimers making them a strong force to be held with the spindle fibers. This way, this is all about the metaphase. One more significance of metaphase for any person or a scientist to study the chromosomes of any particular species or these chromosomes, they will have to consider the metaphase stage, where they will use some kind of chemical agents for example like colchicine, that is to arrest the movement of chromosomes in a equatorial plane. By arresting them, the chromosomes will remain in the same stage, and where the chromosomes size, then the shape, and also the nature of the chromosomes could be studied well. That's what we call it as karyotyping. That's what karyotyping. Karyotyping is done only during the metaphase, because all the chromosomes that are present are very much conspicuously visible under the simple compound microscope. Okay, and once the metaphase is progressed, that will enter the third phase, that is called as Anaphase. Ana means up ಅಂತಂದ್ರೆ movement of the chromosomes. But here I want to tell you exactly how this happens. Here each chromosome, as you know, as you see, here are two chromatids. Okay, and exactly at the center of the centromere there is a splitting process begins, so that two chromatids with half of the centromere and kinetochore, and they begin to get separated because of the pulling action of the spindle fibers. Spindle fibers are originated from one pair of centrioles. They are called as centrosomes. Okay, and because of their pulling action, sometimes they contract, they extend. Because of that reason, chromosomes are formed with a definite shape. They may appear with either U shape or J shape or V shape, like that. Here you can see it very well. See, if you consider this chromosome which is separated with splitted independent centromere, it appears like, look at it, it is a V shape. Here it is a V shape. Here it is U shape. Here it is a J shape. Like that, shape of the chromosomes appear like either J shape or U shape or V shape because of the contraction and relaxation of these spindle fibers. However, equally the chromosomes are now pulled to their respective poles or to the opposite poles in this way. Finally, they begin to repel from each other, they begin to move away from each other, and equal set, look at it, 1 2 3 4, 1 2 3 4, so that here 4 chromosomes, here 4 chromosomes, whereas each chromosome with its own centromere with kinetochore, and it is the same with the remaining all chromosomes, so that upper one set, lower one set, which are initially duplicated, got separated during Anaphase, and finally in the late Anaphase we find very clearly that there is a clear division of the number of chromosomes equally to the respective opposite ends or the opposite poles of a cell. Okay, and once the Anaphase is progressed and completed, now it gets ready to undergo this telophase. One more thing I forgot to mention: what is that force, that driving force making these chromosomes moving away from each other? That is because of one thing. Before they get repelled from each other, there is accumulation of more and more amount of calcium here. Calcium vesicles come and accumulate around the spindle apparatus, and there is release of calcium ions, and these calcium ions enable the spindle fibers to undergo contraction and relaxation frequently, so that the chromosomes begin to get separated, because each chromosome with the two chromatids, and they get separated with equal number, and finally they attain their respective position in a cell. So that's all about Anaphase, and now the beginning of telophase. So initiation of the telophase is indicated by the formation, by the formation of a furrow. That furrow is said to be like what? So that is furrow is said to be like a plasma membrane puckering and furrowing nature of a plasma membrane. Before that, I want to tell you one more thing. During telophase, exactly what all happens? What all is happened during prophase? During prophase is reversed here during the telophase. What is that? I said very beginning of prophase, there is condensation of chromosomes. Here it is decondensation of chromosomes, means heterochromatin gets transformed or converted into euchromatin. Second thing, reappearance of nucleolus, but there is a disappearance. Here it is a reappearance of nucleolus. Then a third event, that is a reappearance of a nuclear membrane or nuclear envelope. This way there is a clear-cut formation of two daughter nuclei. When there is formation of two daughter nuclei with a single set of centromeres, then it is a clear indication that the process of telophase is over. Okay, and once the telophase is over, we could say that the first step of mitosis is over. What is it? That is karyokinesis is over. Okay, karyokinesis, sooner it is completed, followed by the cytokinesis. Cytokinesis also I can explain with the same diagram. As I said, the division of a cytoplasm around each daughter nucleus is possible by the activities exhibited by the plasma membrane. Roughly at the center of a parental cell, there is a formation of groove, and that formation of groove is called as furrowing. At the same time, puckering moment of the plasma membrane, and this furrow becomes deeper and deeper. Exactly where does it occur? If you ask me a question, exactly in the middle, that is said to be metaphase plate, that is the equatorial plane. Metaphase plate is the place where exactly the cell membrane or plasma membrane gets cleaved, gets furrowed, gets puckered, and finally there is a complete separation of cytoplasm with this nucleus from the cytoplasm of this nucleus, and formation of two identical daughter cells. Whatever you see in this daughter cell must be the same with this. Said to be like they are the replica. They are the replica of the parental dividing cell. This way, mitosis is the way for animal cell or a plant cell to produce its own identical replicas or the daughter cells. Once the daughter cells are formed, these two daughter cells may get ready to enter the interphase, and the process, you know that if these daughter cells are supposed to get differentiated, they do not enter the interphase. If they continue to undergo the growth, definitely they get entered into the interphase, that is a G1 phase. Okay, so that completes the mitosis in case of animal cell. Then you may ask the question that is it the same in case of plant cell? Definitely no. Why? Because in plant cell, first and the foremost difference is that there is no formation of spindle apparatus or mitotic apparatus, no appearance of the spindle fibers, number one. And secondly, the cytokinesis is something different. Why? Because the cell, plant cell, is having a cell wall which is rigid, chitin or pectin, something like that, and that doesn't allow to make a kind of furrow. Instead, there is a formation of a cell plate, and that cell plate formation makes the plant parental cell to divide into two daughter cells. However, keep it apart and let us stick on to the animal cell. How does it undergo mitosis so as to undergo the duplication in their number by producing two daughter cells each time? That's all about mitosis. What all we studied in this session? Firstly, the kind of cells present in multicellular organisms body. Secondly, what is that? Significance. Then the type of cell divisions as mitosis, meiosis, and one more amitosis, but it is not significant here to study. And according to syllabus, mitosis we studied. Then thirdly, the detailed account of mitosis, which progresses through the successive 4 stages like prophase to telophase to complete karyokinesis, and division of cytoplasm to complete the cytokinesis. This way, mitotic division is accomplished with their respective molecular events. Okay, I hope you will understand very clearly regarding how exactly cell undergoes mitotic division. Okay, and at the end, I want to request you people that after viewing or watching this lecture video, please put some points, some kind of inputs, whether it is really understandable by you or not. If there are any kind of suggestions to modify our teaching method, yes, I am ready to accept your suggestions. Please put your, not like it is comments box, it is like somewhat inputs. Yes, if I find or if I get information from you like you are following the lecture video, that is fantastic and that is fine, and I will continue the lecture about meiosis in my next session. Thank you very much.